Welcome back. So we are discussing some of the notation and new terms in the basic model for dynamics interacting species, where virus is interacting with CD4 cells. And then we would like to code this model and obtain numerical solutions. So we'd like to compute some simulations of the model, and to do that, we're going to use R. Okay. So let's write a couple of things here because turns out that we're going to have some terms that are equivalent. So in R, we have what we've been writing so far, which is called the mathematical notation. Okay. So as mathematical notation, we have been writing the state variables, and the state variables that we're using right now are t, I, and v, right? Each of them denotes um, one type. T is an infected CD4 cell, I is infected, and V is virus. But when we code these up, if we agree that we're going to use the X notation here in the R code, okay, then we're going to write something that looks like X, and we're just going to refer to this as X1, the first component of X, and I would be denoted in the code as X2, and V will be denoted as X3. So we're basically assuming that there's a vector in terms of the R code notation, a vector X that stores the state variables, and so we use X1 for T, X2 for I, and X3 for V. I'm telling you this because I am about to show you some of the numerical solutions, and I also want to briefly show you some code that would be very familiar to you, with the intention that you would um, replicate this code. So this is um, display here of some of the commands. What we want to do is we want to compute numerical solutions of this model. So we want to compute numerical solutions for um, the population of target cells, for the population of uh, target and uninfected, the population of target and infected cells, and the population of virus. So what we need are values of the parameters of those constants that appear in the model, and we also need values of the initial conditions, and we need to specify for how many time points we want to compute this numerical solution. So this is something that we are very familiar with, solving computing numerical solutions of models specified by ordinary differential equations. And so in R, right, one of the things that you use is this package called, it's a library called DESolve. And one of the functions that is accessible in this library is called LSOTA. Okay, so here is a sample of code, and here you can see how we're making two calls to this function LSOTA. Okay, um, and when we call LSOTA, then we need to specify our initial conditions, the time points. We have to write the function that contains the right side of the differential equations. Okay, and then we have to, the last argument here of LSOTA is that we pass on the values of parameters. So we have two calls because we are considering two sets of values of parameters. So let's look at this code briefly. Um, this first portion is what specifies the function. You can see there are three equations here, and you really are paying attention to the right side of these equations. This is where we have that notation for the code in our x1 denotes t, x3 denotes v, the virus. So this x3 times x1, those are this is one of those terms of interaction between virus and healthy cells. and x2 denotes I, the population of infected cells, right? So very simple, three equations, but we have certainly several parameters. This model has 8 parameters, right? So that's the function that contains the equations. In this case, we're assuming we're going to start the population of uninfected cells with 1,000 cells, and we're going to start with one infected cell and one virion, one particle of virus. Okay, and we have two sets of values. You don't have to really pay too much attention, but I have chosen these values for a demo to give you a demonstration, right? So why two sets of values? Okay, so remember that we had talked about in the context of epidemic models about one type of equilibrium in which the population was free of infection, and another type of equilibrium in which the infected population coexisted with the other populations. Well, here we have the same analogy. We have an equilibrium at which all species here, virus, infected cells, and uninfected cells, they all settle at a stable positive level, right? It's like a horizontal asymptote, and that's what we call the endemic equilibrium. And then we have another scenario in which the population is free of infection. Okay, so we have two sets of values, and one will give rise to solutions for each. Each set will give numerical solutions for each case. And then you have those commands about plotting. So when you execute this code, this is the first these are the simulations you obtain for that scenario in which you have a stable endemic equilibrium. Here you can see in red is the population of healthy cells, and they are all flattening, right? So they are all settling on these horizontal asymptotic levels, right? So and then in blue and green here you have bad news for that host, right? In blue you have infected cells, and in green you have particles of virus. So what's happening here is that these three species are coexisting with each other, yet the virus seems to outcompete the number of target cells. So if you these values of parameters are artificial. They are at this time we're just trying to get a sense of what the model tells us. But this, if we were going to interpret this is telling us that if there is a uh, race here, some type of competition, maybe not explicitly, but there is an interaction between virus and target cells, and the amount of virus according to this simulation is a lot significantly, well, it is larger than the amount of healthy cells. So you could expect this is not good news for that host, okay? And then the other scenario is that scenario in which the disease is not able to establish itself. In that case, we have the same type of initial conditions, so the population of uninfected cells settles at this positive, uh, positive equilibrium level. However, the virus and the infected cells fail to establish themselves. And so you can see here, while this starts with one of each, right? And then after some time steps, this is going to values that are 10 to the minus 1. And if we were going to run the simulation here longer in time, we will see that these values are approaching 0. Asymptotically, maybe not too fast, but they are certainly approaching numbers that are significantly orders and orders of magnitude different than the values here for the population of uninfected cells. So what we have seen for this model, for this basic model, is that in terms of dynamics, right, we have one qualitative scenario in which we have the endemic equilibrium. Okay, that's what this stands for, endemic equilibrium. So uh, what we'll see next is what happens in the presence, what happens to the model in the presence of therapy, right? What happens if there is therapy? So let's say that this model is going to be corrected or influenced or adjusted to take into account therapy. So we're going to make some changes to the mathematical equations. But in terms of the dynamics, what we would like to see is also a change in dynamics, right? And so the change we will see, hopefully for the best, is that we'd like to eradicate. This is a scenario in which the population of virus and population of infected cells are established, right? They coexist at positive levels at equilibrium. So we actually, the therapy hopefully is going to get rid of that, and the therapy is going to try to move the system, perturb the system out of the endemic equilibrium into the so-called disease-free equilibrium. So the therapy wants to bring some, hopefully the system, bring some light. Remember, disease-free equilibrium is an area in which population of virus is going to go to zero, and population of infected cells is also be going to zero. Okay, and so this is kind of the goal. Hopefully, if the therapy is anywhere, you know, close to effective, then this translates into the dynamics, having two dynamic scenarios, and kind of forcing or perturbing the system out of the stability for the endemic equilibrium, hopefully into a basin of stability for the disease-free equilibrium. Thank you.